Good morning, and welcome to the National Invasive Species Awareness Week webinar series hosted by the Southern IPM Center and sponsored by University of Florida, Texas AgriLife Extension, Auburn University, and University of Georgia Center for Invasive Species and Ecosystem Health. Today we are proud to present Carrie Harmon talking about the art of detection, the importance of accuracy in identification and management.
I appreciate this opportunity to speak during National Invasive Species Awareness Week. This has been a lot of fun for me to think about invasive species beyond pathology. I'm a plant pathologist at the University of Florida, so to get to think about invasive species in a much wider setting was a lot of fun for me. So I hope that we have a good conversation at the end. I'll take questions towards the end since I don't see the chat screen at this point and probably won't. But I will be happy to take your questions at the very end. So I know that Joe will be keeping track of your questions in the queue. So what does this fish, this cute little fish, have to do with a hurricane and kudzu and soybean rust? Well, many of you have seen kudzu, maybe not looking like Thor, but many of you have seen kudzu, and probably quite a few of you may have seen soybean rust or certainly have heard of it. I would bet that most of you have not seen a striped knife jaw. This little guy, this tourist, was normally native to Japan and was found in a crab trap just off the coast of Oregon just this week. He's now in quarantine because, just like the kudzu, which now is a host to an invasive disease, soybean rust, he may have parasites or pathogens that could be a problem, little hitchhikers, that could be a problem for our native fish species. Additionally, we don't know if there are other striped knife jaws, could he procreate and then have future problems pushing out some of our native species. So really, when I think about invasive species, really most of these stories are stories about unintended consequences, including the hitchhikers that come along. So I am a plant pathologist. I talk about microbes. I really love fungi, viruses, bacteria. I think the stuff we see in the microscope is super cool. I'm the person that goes to the botanic garden and is looking at the spots on the leaves. I appreciate the flowers, but those spots on the leaves and that wilt over there, oh man, that's my thing. So I'm a little bit weird, but this is what I do. There are folks that also do the same kind of thing for weeds and bugs and snails and snakes and fish and pigs and all kinds of things. But before I get too excited about all of that, I want to define things just a little bit. And we'll do this in a couple of places where I'll talk about words that in the media tend to be used interchangeably but really are not. One of them is the word exotic. Exotic does not necessarily mean invasive. We have plenty of crops that are grown that are exotic, that were introduced to the U.S., that we grow quite successfully, and that are not invasive. Invasive in our terminology tends to indicate that the introduction of whatever the thing is going to cause some harm and leave us worse off than we were before the thing came in. So at the lower left, some of you may have heard some new interesting things about the giant African snails in Florida. They get huge, like the size of my head, which is really big for a snail. You can just imagine. And the really cool thing, two cool things about this giant African snail, not cool, is that one, they eat stucco. They literally eat houses, which is what these two snails are talking about. And two, they can harbor parasites, hitchhiker number one, that can harbor pathogens, hitchhiker number two, that can actually really harm, cause meningitis and other things in humans and bring about actual death, which would be probably the worst side effect I can think of for a snail. So you don't want to be handling snails, you don't want to eat snails or frogs or other amphibians, and for sure you don't want to be eating these giant African snails. They were probably brought in as a food source. There are plenty of folks that eat snails, escargot, and then other ethnicities that find these a real delicacy and even a staple. But these are doing some real damage in Florida. One small piece is that they're eating the stucco off of houses. Another one that some of you may be familiar with, if you're anglers, would be the northern snakehead. I've seen this fish in action. It is horrifying. It's got fangs, it's got real teeth, it's aggressive. It is ruining fishing in lakes up north. It's incredibly prolific. It's a really good survivor. Unfortunately, a lot of our invaders, that's part of the deal, is they have figured out something in their evolutionary pathway has given them an advantage over our natives, and therefore they clear out. Another one you may have heard of, and you've probably even seen videos of, is the battle that's happening in the Everglades between the Florida alligators, our native alligators. Go Gators, by the way. I'm from the University of Florida, right? So go Gators. Fighting off the Burmese pythons, which were probably introduced to the Everglades as pets that people could no longer keep, or escapees. More than likely, they've been introduced as pets. No longer pets, and the two are duking it out, and really we're not sure who's going to win. It's not looking so good for the gators, though. So, away from some of the really fun pictures and onto an invasion curve. Now, the National Invasive Species Council talks about prevention being the first line of defense. Probably won't see me now, but that's okay. The important stuff is that you see what's on the screen. Prevention is the first line of defense, though prevention is not going to keep everything out. And to be honest, prevention really is like stemming the tide to the best of our ability. Early detection and rapid response is where a lot of our education is focused, so that we can increase the likelihood that invasions, once they begin, can be halted and hopefully even eradicated. I mean, we can get rid of the pest once it's been introduced. Once a species becomes widely established, basically we figured out that there is no way to contain it. Eradication possibilities are over. The only action at that point is what we would call an extension we call management or mitigation of the negative impacts. So on our invasion curve, we prefer to be way down here in prevention, where there's no pathogen yet, and the costs to do prevention have been shown in study after study to be lower than the costs of eradication, containment, or certainly management. The longer something gets to grow up, this curve could just as easily be shown to be the population curve of an invading organism. The control costs go up as well. I don't like to use because we so rarely have control, but we do our best to manage. So prevention. There are lots of really good programs about prevention, and one of the ones that I'm familiar with in Florida and have been interacting with is Don't Pack a Pest. Some of you may, if you've been on a cruise or have come in through one of the airports in Florida, you may have seen our little beagle dogs, or you may have seen posters about Don't Pack a Pest. Our Florida Department of Ag is the group that's behind this, and they've done a really nice job of making a friendly notice to people. Please don't bring stuff in that might be harboring some hitchhikers, because you are endangering our native and economic and agricultural products. So you can check, should I bring it? Can I bring it? And then there are people and these cute little dogs at the airports and the shipping ports to look for hitchhikers that might be on products that are being brought in that are banned. If you look up how to prevent the spread of invasives, whatever it is that you're interested in, whether it's weeds or aquatic plants or feral pigs or whatever your invasive du jour might be, if you search that and prevention, you will come up with websites like this. This is from our University of Florida Center for Aquatic and Invasive Plants, and it'll tell you, what do you need to know about invasive aquatic plants? What's the damage they cause? If you've been out on a pond or a lake that's covered over with water hyacinth or some of the other nasties, you already know this. But the nice thing is we can spread that education to our neighbors and to our friends and other folks that we see along the way, and try to mitigate some of the damage and even prevent if we can. Prevention really is the least expensive and most environmentally sustainable way to go about this. Early detection is what happens when we can't prevent it, and I will say that early detection is actually something that we're going to need all the way along, right through management. If we can't find it, we don't know where it is or how much of it there is, well, then we can't be anywhere in the realm of eradication or containment. If we haven't figured out how far it's spread. But even after eradication and containment are no longer possible, and we're in regular management of something that's here every year or every couple of years, we still need to know where it is. And knowing where it is allows us to apply management in a proactive and efficient manner to minimize off-target consequences and keep down waste. You don't want to be spraying if you don't have to. We'll talk a little bit more of that in a few minutes. But it relies on a knowledgeable network of eyes, what we call first detectors or first responders. Really, it's the eyes and boots on the ground. And there are two organizations that I work with extensively. One would be the NPDN's First Detectors at firstdetector.org. Lots of information there on up-to-date modules or trainings that you can go through online. You can get a certificate or not, or just learn about new things. That's kept relatively up to date within the year, I'd say, or even a couple of months of an introduction, or just knowing that something is potentially going to cause a problem if it were to be introduced. Another place you can look is sentinelplantnetwork.org. Now, obviously, these are both plant-based. I'm a plant pathologist, so I tend to work with the plant stuff, but there are plenty of these out that are not plant-based. One of the places I go to often for images, for information, to find out about new things is invasive.org. The group that is sponsoring this webinar, helping to get it online and allowing me to present it from a place that is far away from my home desk. I'm actually at another meeting and I've stepped out just to give this webinar, which is awesome. The support that a group like that can do in technology, as well as things like just finding us a meeting place for all the information. So this is one of my first places to go for invasives, whether it's pathogens or bugs or weeds or the other stuff, pythons and feral pigs and that kind of thing. A lot of information here and a lot of good ways to connect to all the other resources that are out there. So this is my first place to go, sort of my one-stop shop. I'm in the business of diagnosis. I run the Plant Diagnostic Center at the University of Florida. I'm trained to do plant disease diagnosis. So this is my bread and butter. This is what I love, and this is one of the things that I think is of paramount importance when we're talking about managing invasive species. If you don't know what it is, you can't figure out how to manage it, detect it, eradicate it. You have to know what you're dealing with. So identification and diagnosis are crucial to detection, eradication, management, the whole 9 yards. They're a little bit different, but they're really the same thing. Identification can be using tools to just correctly name and identify. There you go, defining something with its definition. And diagnosis is a little bit more. Plants are our patients and they don't talk. So something that a plant can do to tell me that it's sick might be the same thing that it does to tell me that it just needs water or it's been overwatered, a wilt. So diagnosis means putting together lots of clues to come up with a diagnosis and then an actual causal organism, which would be the identification side. So why does it matter, really? Why does it matter if we identify it for sure? If we think there might be a disease, well, why don't we just go to the store and pick up a chemical and spray it out and see if it works? The spray and pray method. Or a herbicide. If we think we've got something in our lawn we don't want to see, why don't we just kill it? Here's a case in point. This is a very new disease. Well, now I've just given it away. This is a new disease in Florida that we're dealing with. This is a dead St. Augustine grass lawn in South Florida. This time of year, the lawns look like this all over the country, but they should not look like this in South Florida. They should be green. This one is decidedly not green. So some of the possibilities that would cause, the organisms that could cause a dead lawn like this, aside from frost or a chemical, something abiotic, some of the biotic things that could do this would be what you see to your right and what you see to your left. What you see to your right is a nematode. The nematodes can basically kill the roots of the St. Augustine grass plants, and then the plant dies. It can't take up water, it can't take up nutrients, it starts to look yellow, like it has a nutrient deficiency. Applying a fertilizer won't help, applying a fungicide won't help. If it's a nematode invasion, you need a nematicide, or you need to correct the underlying problem. What you see on your left is the new disease that I was talking about, and it's a virus. Those long stringy particles are actually EM images of an actual virus particle for a podovirus that causes these streaks, but eventually causes necrosis or death of the plants themselves. In this case, applying a fungicide still won't help, even though it's a disease, because a fungicide does not treat a virus. This can be extended to pretty much everything else that we talk about with invasives. We will need to know what we're dealing with and what the options are for dealing with it. So if we're talking about invasive animals, we might be looking at the behaviors of those animals and therefore what would be the reproductive management or trapping potential for them. For weeds, we might be looking at habitat and chemical resistance as far as herbicides. We might look at behaviors in host plants for insects And for the insects, weeds, and animals, we might also be looking at what they might vector, what would they bring with them. Animals might have weed seeds attached to their fur. Weeds might have pathogens or insects hanging out on them that we don't want. And then the insects might have pathogens that they're moving around from plant to plant. And then, of course, there's the part that I do, which is I look at the conducive environments or host plants, and then the application of effective chemistry or other management options for those pathogens. On the lower left, we're looking at feral pig damage in a cemetery. So I didn't put up pictures of the pigs, but if you know what the damage is that these animals can cause, you understand why they are an invasive. They're definitely leaving things worse off than they were before the invasive came in. And on the right, we've got herbicide-resistant Palmer amaranth, which I think is also sometimes called pigweed. So I thought that was kind of a nice, both kinds there. And then management versus prevention. I talked a lot about prevention being the least expensive way to go. It's better for the environment, and that is for sure. Management rarely results in complete eradication or control. Some of our management options will destroy non-target organisms. For instance, a herbicide that we're spraying on a weed could also impact the crop plants that we need to be protecting. Management assumes some tolerable level of the organism will probably remain. What that tolerance is and how much we can deal with is something that usually our research friends will tell us and help us with. And sometimes we don't want any of it, in which case we really need to be trying for eradication. Integrated management, so IPM, and it is the Southern IPM Center that sponsored this webinar series, indicates an effort to utilize lots of methods to decrease the target pest, whatever that is. So it might be chemical, it might be cultural. There are lots of ways to manage. And in the end, we're going with an ounce of prevention is worth a pound of cure. Cure being a four-letter word in my vocabulary because we rarely can cure a plant of an affliction, of a sickness, a disease. We can stop it in its tracks, but we don't actually cure it because they don't have an active immune system like you and I do. So we can be proactive or we can be reactive. The upper left, playcleango.org, is a great website, a good organization for those who are going out and taking part in recreation, whether it's in national parks, that kind of thing. The idea is that you don't want to bring anything in and you don't want to bring anything out. We're not just talking about packing your trash. We're also talking about what's on your boots. If you went hiking last week in a park on the eastern part of the state and you're going to go hiking in the western part of the state this week, make sure that there's no mud in your boots that might bring weed seeds in. Burn local or certified firewood. We've moved several pests around. We know that we have moved them around. In firewood, wood-boring beetles, diseases that have moved around on firewood. Along the same lines, use local or weed-free hay to feed your horses if you're doing an equestrian activity. You don't want to be introducing new weeds to a new place. Stay on the trails. While we don't want to have to treat for, say, weeds on a trail, at least if we're walking along a known trail, we can look for them there and treat them on the spot. If you go off the trail, they're going to be a lot harder to find until they've probably escaped the potential for eradication and containment. And then before you leave, think about what you're taking out. Take pictures, right? Not anything out of the forest, but also don't take any mud home. Don't take any weed seeds. Take a look at the cuff of your jeans. Take a look at the boot, the tracks in your boots, and try not to take anything out. The folks you see with the backpack sprayers, that brigade there, they are going to spot treat for weeds that escaped. The same kind of weed issue exists in the aquatic realm. You really don't want to be pulling your trailer out of a pond and have that kind of a hairiness on it. If you can, before launching, make sure you're not putting anything in the water, and when you leave, don't take anything with you. None of us want a lake that looks like the lower right-hand picture, which is completely covered with 3 different types of aquatic weeds, one of which is nearly impossible to control. So, pop quiz. Which one of these is an invasive disease? You're looking at a rhododendron plant. We've got a leaf spot, leaf blight issue, and if you looked at the one on the left and said, this has got to be it, bing, 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 you get a gold star for today. That tip burn represents the symptoms of Phytophthora ramorum, which is the organism that causes sudden oak death, and ramorum blight, which has killed a tremendous number of coast live oaks and other vegetation on the west coast, and we are trying to keep it out of establishment from the east coast and the rest of the country. The picture on the right is simply burn. It's actually drought and heat stress on a rhododendron, and would not be treated like we would treat the plant on the left. The plant on the left needs probably application of fungicide, and then we also need to make sure that it's not on a shipment of plants that's moving across the country. Another pop quiz: if you're more into bugs than you are into pathogens, can you spot the emerald ash borer? And then I want you to take your computer screen and I want you to shake it. Could you spot the emerald ash borer then? If you said top left, then ding, 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 you get a gold star again. The issue with spotting something like this little tiny insect is that there are lots of things that could look like it, and they're on the move. Diseases stay in place because the plants stay in place, but insects, animals, they're on the move, and they're a lot harder to pin down. So in this case, if you can capture it, if you can take pictures of it, those are the things that you're going to need later to confirm you've got it. And the people you're going to bring those to, the folks in the diagnostic labs and your identifiers. Call us your friends in the invasive species business. Land-grant universities like the University of Florida, where I work, and state departments of agriculture all have labs for the identification of invasives and pests of all kinds. You can find out how to get to your local one by going to npdn.org, and you'll find the one in your state closest to you. These labs are usually connected to specialists like myself who research and recommend management appropriate for the local area. So the chemicals that would be appropriate, the changes to cultural methods that would be appropriate for your system in your state. The easiest way to find them, search your state plus the word extension, or you can even go to eextension.org and just type your question right in and have somebody from anywhere in the country answer your question there. So this is what extension looks like. Solutions for Your Life is what our extension system is called at the University of Florida. And on the page just yesterday, Managing Invasive Species. This is an important topic in extension, and we've got the tools to do it. Think you've got something new? Tell us all about it. Don't keep it a secret. Talk to your local extension folks, your IPM specialists. Often they're one and the same, not always. Look up your local Department of Agriculture or Invasive Species Council. These folks would love to know if you've spotted something, and if they already know about it in that area, they'd probably love to have your help in managing it. Get pictures, location information, collect a specimen if you can. Those are the things that you're going to need to back it up. And of course, because this is 2015, there's probably an app for that. One of those, or two of those, would be, I've got one in Florida, in the Everglades. This is a very cool app where you can report what you've found, and it can be confirmed by image, by a specialist, and then we can map these things through EdMaps and several other mapping features through other websites, so that you know where things are and if you should be on the lookout for something. So one more time, can you spot the emerald ash borer this time? These are things that you might have to worry about. Lots of other bugs that might look when they're all flying, like the emerald ash borer. Obviously, the one that we've circled in red is the one you'd be worried about. You might not confuse it with the annual cicada, which is off to the right, but if you didn't know what the emerald ash borer looked like and how big or small, in this case it was, and you saw the cicada, you still might be concerned that you've got something that needs to be reported. Can you spot it now? So at this point, I appreciate your attention. I will take any questions you have. Thank you for letting me give you some information about identification, and I hope that from here on out, if you're thinking about management or eradication, the little voice in your head says identify, identify, identify.
Excellent. We have plenty of time for questions. If you have any questions, you can raise your hand, and we can try to unmute your audio and have you ask your question that way. Or there is, on the bottom next to the chat box, a place where you can enter your question online through GoToWebinar. I actually have a question for you, Carrie, about what type of things I could bring to the diagnostic lab. Could I bring, here's a snake I found?
Well, the answer really is going to be yes in general. We can always direct you to the right place. My diagnostic lab deals with plant diseases, but very often somebody will walk in with a plant issue and it needs to go to the insect identification lab, it needs to go to the nematode assay lab. There are lots of labs that are fully integrated, so you may walk in with almost anything. I would say if the snake is alive, it probably better be contained. And you don't want to be moving them long distances, anything that you're bringing, because we don't want to be spreading this as we're trying to identify it. So my first line of defense tends to be take it first to your local extension agent. There's one in Florida, there's one in every county. Lots of states still have very strong extension systems. The first place you might want to go is your local extension person and talk to them about what you've found. If you've got a giant Burmese python in the Everglades, they're probably not going to want you to bring it in, but they might. Call your local extension folks and ask them what the procedure should be for what it is that you're concerned about. That would probably be the first place I would go. Does that answer the question, or is that...
That answers my question. I now know that if I bring in a giant python to the lab, you'll be okay with that.
I hope it's in a cage, and then I'm going to send you over to our animal people.
We do have a question from Chris Dionigi. As you look across the different subject areas, entomology, plant pathology, plants, is there one that stands out as having gaps in capacity? I imagine gaps in capacity for diagnostics? Yes.
Well, absolutely. I would say that there are gaps probably for identifying all of them. Each discipline is probably going to say that there's simply not enough manpower to identify them all and get all the work done, and that's probably true. I would say, and I can speak best for my own field, plant pathology, that the identification of a causal organism for a plant disease is a lot harder, because if you take a picture of a wilted plant, I can't tell you what's causing that wilt until you bring it into the lab, which means that we have a bottleneck of sorts at the laboratory. Whereas you could send our entomologist a really good set of pictures of an insect, and they might be able to identify it right then and there, which kind of spreads out the capability. As I was surveying across everything, I would still say, and I'm sure that it's my professional bias, that diseases would still probably be a place that we're going to continue to need to train people to help us and to be diagnosticians, as well as to be on the lookout for things that could be a problem, simply because there are so many things that can cause the same symptom. Any other questions?
Yep, they're slowly trickling in. So do you see prominence in newer molecular approaches to identification? I guess promise is what he was saying, not prominence.
Do I see promise in molecular identification? I do, and we use it. We are increasingly using it for the identification of pathogens. It is starting to be used more in nematode identification, so the things that we can easily identify by looking at with our eyes, whether through a microscope or macroscopically, are going to be a little less of an issue there, unless it's a species issue. We need to know, is it this species or that species? And then molecular diagnostics would be very useful there. It continues to be very useful in plant pathology, and I think that's going to increase, with one major caveat. Molecular diagnostics do not tell us if the organism is alive, if it's infectious. So because we can detect the presence of the DNA of that organism on, say, the outside of a piece of fruit, does it mean that that organism is causing disease? Does it mean that it's going to be an invasive issue? Not necessarily. So as long as we know that there are some caveats to using the newer molecular approaches to identification, and we use them carefully, I think there is real promise there. The other issue with molecular identification is simply the cost and the time involved to get the nucleic acid out, to use, whether it's PCR or we're going straight to sequencing, whatever it is that we're going to use. And then the last piece of that is reference material. You have got to have a positive control to run, and getting positive controlled DNA or RNA for an organism is sometimes really difficult. But if you don't have that, then the value and the validity of the test are decreased. That's a really good question, and I could probably talk for another half hour about that.
And in addition to the molecular tools, there's also a fair amount with ELISA and those tests. How does that work into the triaging stuff for diseases?
Those are a little bit faster, some of them. So we have things that are dipstick, sort of like pea on a stick. You get one line, you don't have anything, 2 lines, it's a boy kind of thing, or in this case, it's Phytophthora. Those are nice and quick, but they are not very specific. And then as far as the other ELISAs, they can be specific or not, depending, but they may take up to 2 days to run, and require some specific equipment and reagents, and they can be a little bit expensive as well. We in the diagnostic lab do what we call triage and best use. So we look at and develop a hypothesis and then apply the best and most appropriate test for the organism we're looking for, or set of organisms, and the client. So if it's a $40 rose bush and we're looking at it, we're pretty sure that that's black spot. I'm not going to run PCR to find out the species of the organism that's probably causing the black spot. I'm going to tell you that it's black spot because I've seen it enough, and we can see under the microscope that it has the signs of being black spot, and that's going to be good enough. If, however, we're looking for lethal yellowing in palm, I'm going to need to run PCR because there's nothing else that I can do to test for that organism besides look at the symptoms, which again are not diagnostic.
Excellent. Everyone is very happy with your answers there. Thank you very much for that. I see no additional questions at this time. So given that, I'd like to thank you all for attending this webinar as part of the National Invasive Species Awareness Week. The recording of this webinar and all of the other ones that we've done are going to be available online at youtube.com/southernipmcenter.